This lecture is going to be on nucleic acids and viruses. The nucleic acids are biopolymers and they contain monomeric units called as the nucleotides. And you have already seen many a time the DNA structure, which is a double helix, and each of those units, what we represent as letters, these are the nucleotide units. So just like how we had in case of carbohydrates, if you have multiple monomeric units of saccharides joined together, it becomes a polysaccharide. In case of nucleic acids, we also have what are called as polynucleotides, which is a chain of nucleotide monomeric units. Now let's look at the structure of a mononucleotide. It has three parts to it. One is the phosphate group, the second one is a sugar, and the third one is a nitrogenous base. All three of them together make a nucleotide unit. Now, if you were to have just the new nitrogenous base and the sugar, this is called as a nucleoside unit. Side meaning sugar containing with nitrogenous base. Now, the phosphate group here, the phosphate group is going to be the charged part of the nucleotide. And you can see that usually the phosphate group is PO4, and when it is connected, it will have a double bonded oxygen, one oxygen with a negative charge, and O phosphate like that. And that is attached to a sugar. And if you remember from the carbohydrates, we talked about sugars that are seen in case of nucleic acids. And these are the ribose sugar and the deoxyribose sugars. These two sugars, they are 5-carbon sugars, and that's why these are also called as aldopentoses. So we have carbon number 1, 2, 3, 4, and 5 carbons. And the aldehydic group is now cyclized into that ring structure that we see here. The aldo meaning it contains an aldehydic group as the functional group, and pentose meaning 5 carbons in the sugar. The name also tells us what is the difference between the ribose and the deoxyribose. Deoxy meaning without the oxygen or the OH group. And specifically, if you look at carbon number 2 in ribose versus carbon number 2 in case of deoxyribose, the OH is replaced with hydrogen, which is why it is called as a deoxyribose sugar. Now, this small little difference affects its stability. DNA without the oxygen is much more stable compared to the RNA when it has the 2'. This is called as the 2' OH group on the ribose sugar. Riboses are present in case of RNA, whereas deoxyribose is present in case of DNA. So this is the sugar unit in the nucleotide monomer. Now the nitrogenous base contains multi-nitrogen containing ring systems. If we have a 2-ring system, these are called as the purines. You can see there is a 5-membered ring on here, 5-membered ring, and a 6-membered ring fused together. The two examples that you will see as purines will be adenine and guanine. And the major difference between adenine and guanine is what is attached on the 6-membered ring, and these are responsible for forming hydrogen bonding interactions when there is complementarity between bases. The second set of nitrogenous base, these are called as pyrimidines, and the major difference between purines and pyrimidines is that it contains only one ring in each of them. Again, there are at least 2 nitrogens in these molecules, and the two nitrogens in the ring for sure in all of them. That is the common. What is going to be different is what is attached in between those structures right there, and also the methyl group in thymine. So in that way, you can, if you compare thymine to uracil, you can see that they have exactly the same structure except for the methyl group here. And that's why you can also refer to thymine as 5-methyl uracil derivative. So the nitrogenous bases are classified into purines and pyrimidines. Purines have adenine and guanine, and pyrimidines we have thymine, cytosine, and uracil. One of the most important mononucleotide is the ATP, which is used as the energy currency of the cell. So it contains adenine as the nitrogenous base and the ribose sugar as the sugar part of it, and it contains 3 phosphate groups which are shown in that phosphate linkage. The sugar has carbons numbered from 1 through 5. Nitrogenous base is attached to the carbon number 1, whereas the phosphate groups are attached to carbon number 5 here. And 3 phosphates are in a linear chain, and each of them having negative charges. Now, this is the most important molecule, and nature has chosen this to be the energy currency, such that hydrolysis of these phosphate groups leads to release of energy. So when adenosine triphosphate gets converted to adenosine diphosphate, the gamma, these are called as the alpha, beta, and the gamma phosphate groups. The gamma phosphate group is hydrolyzed whenever ADP is converted to ATP, and that's shown here. So we see the alpha, beta, and the gamma in case of ATP, and when it gets hydrolyzed, the reaction is called as hydrolysis. The bond breaks between the beta and the gamma. That bond breaks right there, and that leads to the release of energy. Now there are only 2 phosphate groups remaining, and hence it is called adenosine diphosphate. If there is a need and specific reactions, sometimes some reactions do require a lot more energy. So ATP can be broken down into AMP, which is monophosphate, where it has got only 1 phosphate group remaining on the molecule. And those reactions are going to be specific for certain molecules or certain metabolic pathways. If you remember hydrolysis is breaking a bond and using water as a reagent here. So one of them will end up having the hydrogen and the other one will have the hydroxyl group when the group gets divided there. There are 2 basic nucleotides that are formed as polymeric forms, the DNA, of course, and the RNA molecules. There are multiple types of DNA. We have, like, DNA can exist as predominantly it is in the double helical form, but in some cases they can form the telomeres, which is quadruplex form, and so on and so forth. RNA also, there are at least 3 different types of RNA, the mRNA, the tRNA, and the rRNA. mRNA stands for messenger RNA, which gets the message from the DNA into the cytoplasm. Transfer RNA is the tRNA that is involved in transferring the amino acids onto the ribosome. And R is going to be the ribosomal RNA, which is part of the ribosomal machinery. Now, one of the major differences between the DNA and RNA is that DNA exists predominantly in a double helical structure, whereas RNAs are single-stranded and they don't generally form helix. And because of the 2' hydroxyl group on the sugar, so this has a 2' hydroxyl group on the ribose sugar, and that makes it very susceptible for hydrolysis. And there are also a lot of enzymes called nucleases which are present, the RNases to be specific. Those will hydrolyze RNases much more rapidly in the cells. So RNAs, they are different from DNA mainly because the role of mRNA or tRNA and RNA is much more temporary compared to the DNA, which is used as a storage of the genetic material that needs to be passed on from one generation to the next generation in an intact manner. Now, at the molecular level, the difference between DNA and RNA, DNA consists of 4 bases, that is A, G, C, and T. A and G are the purines here. C and T are the pyrimidines. In case of RNA, we have AG as the purines still, whereas instead of T, we have uracil as the pyrimidine. So this is one slight change in the bases. Predominantly they contain AGCT in case of DNA and AGCU in case of RNA. And the linkage and everything else is very, very similar. Again, of course, the deoxyribose versus the ribose, but the arrangement of the nucleotides in that the sugar phosphate backbone is used as a linker for the mononucleotides joined together to form a chain in both cases. And so we call this as a sugar phosphate backbone, and the nucleotides, the nitrogenous bases, are facing the other side of the chain. That structure is very similar to both DNA and RNA. In terms of functionality, there is a huge difference. DNA is used to store information, the genetic information coded in the DNA, and that is required for the viability of the organism. So all the coded information as to what sort of proteins needs to be synthesized, when the proteins have to be synthesized, and what should be their function or structure, all of that is encoded on the DNA in the form of those polynucleotides we just talked about. Whereas RNA, these are involved as messengers. They only copy the information from the DNA and transfer it to worker molecules, the proteins inside the cell. And they're also involved in the process itself, transferring the amino acids to make a chain of amino acids, which is again a polypeptide chain, or in the construction of the ribosome subunit itself, which is involved in protein synthesis. In case of the double helix, when one of the strands is going to be paired with another strand of DNA, one polynucleotide strand here that is shown on one side, that can be paired with another one, and in the process they twist around each other to form a helix, as shown here. So in each of these strands, you can see that the sugar phosphate backbone forms the string part of it that holds the chain together. And the nucleotides are going to be facing each other, and in the process, the proximity makes them bond with each other through hydrogen bonding interactions. And the hydrogen bonding is also very specific. A will base pair with T, or A will base pair with U. In case of DNA, we have this. In case of RNA, we have AU. And this base pairing is with 2 hydrogen bonds between the nucleotide bases. On the other hand, we have G base pairs with C. In both cases, it's the GC base pair, we call it. And these have triple bonds, hydrogen bonds between the two. So there are 2 hydrogen bonds in AT or AU, whereas there are 3 hydrogen bonds in case of GC base pairs. And the base pairing is such that the bases are almost going to be flat. And that is why you can see that sometimes when we represent this, we represent it as rungs of a ladder. So they were so flat that in fact they look like the rungs of a ladder. And that flat structure is the reason why there is stronger hydrogen bonds which can be linear between the two bases that are facing each other. So the presence of these hydrogen bonding between the base pairs and also what we call as the stacking of the base pairs using the van der Waals interactions or even electrostatic interactions gives very high stability for the double helical structure of DNA. And you can see that the DNA structure is so intact because we are able to extract the DNA from the fossilized dinosaurs or any other animal or plant, and we are able to analyze and study those DNA structures because they're still intact. It's all because of these hydrogen bonding interactions and the base stacking. Now, when I talk about the base stacking, it's the rings have pi bonds. You can see that there are a lot of these p orbitals which are going up and down each of the ring structures. When we talk about the base stacking, it's the interaction between these pi bonds. So there is a p orbital right here. So these guys can interact with, right? They're sort of overlap. So if you have the p orbitals from the top and the p orbitals from the bottom, so you can see some sort of p orbital interaction, and that is called as the pi stacking, pi base stacking interactions that provide additional stability for the duplex structures. So at this point I want to take a small break, and I want to have the in lecture based questions which you need to answer and submit on the Canvas. So please make sure that you write down the answers for these questions on a separate sheet of paper and upload it to the Canvas in the lecture assignment tab. You can pause the video at this point, understand the question, and write down the answer, and then come back to the lecture and continue with it. I will give a few seconds, but I'll continue with the next few slides. All right, I will continue, but please make sure that you have answered those questions before you move on to the ester bonds. There was a mistake on this slide on one of the earlier slides that were posted on the Canvas. Please make sure that these are phosphodiester bonds between the nucleotides. And the way these phosphodiesters are formed is when the phosphate group of one nucleotide interacts with the 3' hydroxyl group of another nucleotide. So you can see here, we have shown that the hydrogen from one of the hydroxyl groups on the ribose and the OH group on the phosphate are going to be removed as a molecule of water. And in the process, the phosphate will be connected to the oxygen of one of the ribose sugars and the CH2 group, which is on the 5th carbon in the next nucleotide. So this is why we call the link between the two nucleotides as the 5' to 3' connection. There is a directionality in the connection that is formed between the nucleotides, and that always runs in the 5' to 3' direction. Just like how we had in case of nuclear proteins, it was from the NH2 terminus to the carboxy terminus. So the nitrogen NH2 on the on the amino group interacts with the carboxylic acid group on another amino acid, and together they form a peptide bond. Here we have the hydroxyl group of one sugar interacting with the phosphate group and thereby form a phosphodiester bond. So this is going to be the phosphodiester, where you can see the phosphate being attached to the two carbons, the 5' carbon as well as the 3' carbon between the two. And this is also called as a dehydration, mainly because we get rid of a water molecule in the process. So now you can see when the two nucleotides are connected, how the phosphate actually links between the sugar groups right there. And this can continue through multiple nucleotides to form long chains of nucleic acids. So the phosphate group is going to be negatively charged and facing on the exterior, and the nitrogenous base will be facing the interior, where they can form the triple hydrogen bond or a double hydrogen bond based on the sequence they're pairing up with on the other strand, if it is a DNA, but they could be single-stranded as in the case of RNA as well. In case of the duplex DNA, we have the two strands running in the opposite direction. If one strand goes from 5' to 3', the other strand will be 5' to 3' direction. And this is when they can have perfect complementarity between the nitrogenous bases, and they end up stacking really well and also forming the linear hydrogen bonds, which are much more stable and add additional stability for the entire structure. So the backbone, the string that is attaching, that is holding the nucleotide chain together, is called as a phosphate deoxyribose backbone. And we have the nitrogenous bases, and that base pairing is again adenine will base pair with thymine with 2 hydrogen bonds, and guanine will base pair with cytosine with 3 hydrogen bonds. So knowing the sequence of one of the DNA strands, we can easily figure out the complementary DNA strand. So let's say, for example, if we were to have a strand which is 5' to 3' running in A, G, T, C, A, T, G. Now we can easily write the complementary, which runs in the opposite direction, 5'. A will base pair with T, G will base pair with C, A, G, G, T, A, C, and we end with the 5' there. So again, you can see that the base pairing between AT will be double, and base pairing between GC will be triple bonded right there. So at this point, I want to have the second question for the in-lecture questionnaire. And you can pause again here, answer this question, and continue once you're done. So the question here is, why are the GC base pairs more stable than the AT base pairs? Now let's look into the organization of the genetic material, which is the DNA structure. So normally in the cells, we refer to them as chromosomes. The humans have 23 chromosome pairs, out of which 22 is autosomal, meaning they are required for required for the cellular activity, and one is used for the sex determination of the cell. So you can see that these chromosomes are heavily condensed structures. They have this sort of a structure that you have seen where it forms like an X sort of a thing and held together by the centromeres at the center. And if you were to expand any particular region of this chromosome, you will end up looking at it as a DNA double helix. And when you separate each of those strands, each strand will contain the sequence that codes for a specific gene, and each gene meaning it is responsible for a protein product. So this leads us to understanding the central dogma of life, where the duplex DNA, which is present inside the nucleus, is going to be having all the information for any particular gene that needs to be converted into a protein. And each gene will be copied individually as an mRNA sequence. So the information from DNA to mRNA, and this process is called as transcription. So mRNA is the transcribed part of it. And that mRNA, once it is synthesized in the nucleus, it goes through a few modifications. And once those modifications are done, the messenger RNA comes out of the nucleus into the cytoplasm, where it will assemble a ribosomal complex, which is involved in the synthesis of proteins. So the ribosomal machinery will bring in the amino acids in the form of tRNA, and then these amino acids are going to be linked to form polypeptide chains on the ribosome. So this process of mRNA being converted into a protein sequence, this is called as translation. So the coded information is translated into proteins in the cell cytoplasm. So DNA being converted to mRNA is called as transcription. And mRNA being translated to protein is again translation. And these two reactions, they form the central dogma of life. And we also know that DNA can replicate so that it can be amplified, or for growth or purposes, or even for passing on from one generation to the next. So when we have the DNA in the form of genes, each gene has a specific set of DNA sequences that are responsible for recognition, and then each sequence which codes for a particular protein is going to be the one which is translated. So the promoter region, this is called as the Tata box, usually contains the Tata box, meaning it contains thymine, adenine, thymine, two adenosines together. Like all of this sort of a rich sequence forms the promoter region, where we have the enzyme polymerase come and bind and copy the information, which is the coded information for the gene. So when you look at the mRNA sequence, it will be the complementary sequence for the 3' to 5' DNA. And the mRNA, once it is synthesized, it will go through modifications in the form of 5' methylation at the guanine, and also the addition of the adenosines poly A tail. So these are to ensure that the mRNA doesn't get degraded, number one, and they also have an address where to go in the cytoplasm in order to decode it into the protein. So whenever the DNA is unmethylated, meaning if the DNA itself it is open, it doesn't have any methyl groups on the DNA, then we say that the DNA is open for transcription or there is active gene expression happening in the cells. However, whenever the cells are in a dormant state, when there is no need for like any of those specific proteins to be synthesized because that metabolic pathway is not in the active path, then these DNA can be methylated. The DNA gets methylated, and this addition of the methyl group on the cytosine, as you can see here, 5-methyl cytosine is going to be the product. Whenever we have the cytosine methylated as this form, then we call it as a methylated DNA, and methylated DNA is called the silent gene, and there is no transcription happening or it's not being converted into a protein. So this is how many a time many of the genes are switched on or off depending on the requirement inside the cells. And this has like become a really hot topic because regulation of gene is all because of these sort of small little chemical changes that are like made on the DNA. So in case of, you can imagine in case of cancer cells, the DNA is not methylated at all because it's getting actively transcribed and translated because the cells are in heavy cell division phase. So these are the different small things that will be useful for designing molecules for therapeutics. How to control gene expression so that we can come up with gene therapy. So the gene therapy based ones, we have certain things which are called as DNA directed therapies where we can design small molecules which can simulate the switched on or the switched off version of the genes, thereby modulating the expressions. Or we can also have RNA directed therapies where we try to target the mRNA, which is the copied information. So either you destroy that or you amplify it depending on the requirement. So even in the DNA directed therapies, we have gene transfer or gene editing. Some of these are still in the nascent phases in that we don't have applicable therapeutics yet. But they're very promising, and we probably will be able to see some of these applied pretty soon in the next decade for sure. The RNA-directed therapies like the siRNA or the antisense oligonucleotide, these were in the process, but we do have a lot of different products that are available in the market. You may have heard of these siRNA, small interfering RNA, that are supposed to inhibit the translation process when the mRNA is going to be converted into a protein, or even antisense oligonucleotides, which go and bind to these mRNA molecules and trap them from being translated. So these are all the, though DNA, it would be an easier way to, it will be a much more targeted way of regulating any of these gene expressions by designing small molecules that can interact with DNA. But nonetheless, it is not easy to design a molecule such that it only affects a particular gene. It might have effect on the other genes, and we have to be really careful as to how we want this to be. What sort of processes do we want it to be affected in a cell? So we will talk about some of these RNA therapies like the mRNA vaccines that are part of our virus chapter. So I just want to introduce the topic of viruses. In the context, we have COVID vaccines and all of that stuff that you probably are aware of. So it's a good thing to understand where this is coming from and how is it applicable in case of therapeutics. Now, viruses, these are tiny biological particles, though they may not be referred to as living organisms. they have the capability of being converted into live organism in any suitable host. So they're basically at the molecular level, they're composed of either DNA or RNA, and that is enclosed in a protein coat. That's the structure of a virus. It's a very basic um, polymer of biopolymer that's just encapsulated in a protein coat. The only requirement is they just cannot replicate on their own. They always rely on a host and the host cell machinery in order to replicate and reproduce or even get propagated. There are a lot of diseases that have been identified as being caused by viruses: the common cold, the coronavirus, the rhinovirus, all of these, like many of those fall into this category. Influenza with its orthomyxovirus, the herpes, the HPV, some sorts of leukemia, and some of the cancers, as well as AIDS. These are all caused by the retrovirus. Hepatitis, mumps, chickenpox, all of these are caused by viruses. So, as I said, the proteins consist of a nucleic acid, which is their genetic material, and usually that is covered with a protein coat. And they have enough information in the DNA that will be responsible for the synthesis of proteins that are necessary for their viability as well as their propagation. In some cases, all viruses will have a protein code that we have, the genome, and some enzymes that are required for the um, particles to be able to um, revive again. But some of the viruses can also have an external envelope, which is usually consisting of a lipid layer, and they also may have some embedded proteins which can act as envelope proteins that is very specific for these types of viruses. Viruses are classified based on the genetic material, either as DNA viruses or RNA viruses. And the final one is the reverse transcribing virus, the retrovirus. These are called as the retroviruses. They need their RNA genetic material need to be converted back into DNA before they can become activated. Some of the examples for the DNA virus are the herpes virus, pox viruses, or adenoviruses. Those are responsible for the common colds and the respiratory diseases. The adenoviruses are the most common ones. Now, the RNA viruses, those cause influenza or the COVID-19 and the SARS. The rhinoviruses, the rhino meaning nose, like those affect the respiratory ones. Poliovirus or Ebola virus is also a type of an RNA virus where the genetic material is made up of RNA. And we have the retroviruses. The classic example is the HIV or even the hepatitis B virus. These they contain an RNA genetic material, but in order for them to be able to propagate, they need to be converted into DNA, integrated into the host, and then they can be multiplied inside the host cell. Because the fact that they can get integrated into the host genome, it is very hard to track them or inactivate them once they are in the host genome, and that is why it becomes really hard to target the HIV or the hepatitis B virus. We don't have any way of selectively attacking that gene because now it is part of the host genome, and then it gets replicated every time the host goes through replication, and sometimes it can trigger to really consequential outcomes where the patient can get severity of the problems drastically increase and affect the overall quality of life. Now, most of the viruses, as I said, they need a host in order to become active or viable. So the viruses enter into the host cell by attaching to the cell membrane. And at that point, the virus coat is going to stay outside. It just gets, it's useful, all of these receptor proteins or anchor proteins, they interact with the receptors of the host cell, latch onto the cell membrane, and then fuse into that and allow their like nucleic acid, the genetic material, to enter into the host cell. So inside the host, the viral DNA gets integrated into the host genome, and anytime the DNA gets replicated, like the host DNA gets replicated, the viral DNA is also going to be replicated. And sometimes the viruses have enough coded information for that to be multiplied many more times, leading to a quick increase in their genetic material. And at the same time, the genetic material of the viruses also contain information to synthesize the protein code. So once the DNA and the protein code are synthesized, they get assembled into the virus particles, and the virus particles can be released when the host cell is going to be ruptured. And that's the reason the host cell dies. But the viruses can go and affect newer and newer particles, and finally they can attack many cells in the body and lead to death in some cases. The COVID virus is something that I wanted to just highlight here. It's slightly different from the rest of the virus, mainly because it not only has the DNA material and RNA genetic material, it also has a protein code, and we have what are called as these proteins that are synthesized on the protein code and embedded into that. We have different types of protein, the E protein, the M protein, and these are heavily studied, and we are trying to get drugs that can actually specifically target these proteins, thereby attacking these virus particles to be destroyed. So one of the common drugs that we use for COVID is called as Paxlovid. You may have heard of this. Or it's used usually as an oral tablet or a capsule. Paxlovid. In fact, it contains these two compounds in some ratio as a mixture of these two drugs. Nirmatrelvir, Trelvir, and Ritonavir. So these two compounds, as you can see, that they contain. Can anybody recognize these functional groups here? We have the NH and CO right here. We have a nitrogen in the ring. We have an oxygen here, and OH here again. So it's a good exercise to identify these functional groups. And you can see that these are big molecules, but nonetheless, these have shown to be reasonably specific for the M protein on the COVID virus. They attack the viruses with the M protein and target them to be destroyed by the host immune system. These are other drugs that have been tested, and they're in the process being approved for oral COVID antiviral agents for treatment of COVID. So the Paxlovid that I was just talking about, the way it works is it has the Paxlovid is going to be specific for the M protein that is produced on the surface. So as soon as the M protein is synthesized by the virus's genetic information, Paxlovid can interact with that, bind to them strongly, and cause the protein to be degraded. The M protein gets degraded because of Paxlovid's interaction with that. It also has an amide linkage, which is why it can integrate and break those molecules. And once the M protein is going to be disintegrated or cleaved, then you don't have enough proteins for the assembly, and the genome replication is not going to be, this process is not going to be happening, and thereby the infection will be subsided. Excuse me. So the COVID vaccine is what we are normally used to. And the way the COVID vaccine has been developed is it's used as the spike protein on the COVID vaccine that has been developed as an antigen. And once this is not producing the same effects, but it has the antigenic properties. So we once the mRNA that's responsible for the spike protein is going to be injected, that enters the human cell. As you can see, the mRNA, which is the sequence for that protein, it's injected into the cell, and once it enters, it's going to release those spike proteins on the cells, and those spike proteins are going to be recognized by the antibodies, and the antibodies get synthesized immediately, and thereby prevent the viruses from multiplying further, or it just mitigates it. They just attack the virus particles before they can become consequential in the body. So this is how the COVID vaccine works. It's an mRNA vaccine. The mRNA is going to be the sequence that is responsible for the spike protein, and that triggers the antibody formation in the body, and thereby it keeps the immune system active enough not to let the increased infection spread around inside the body. So finally, I want to end this lecture by giving you these last two questions on the lecture. What makes viruses to be considered as living, and what makes them to be considered as non-living? So there's always a constant debate as to whether viruses are living organisms or non-living. So I would like to see what you think, and which way do you weigh in. The second thing I want you to think is, why can't we use antibiotics for viruses? We have a lot of antibiotics in the market, but none of them will be active against the viral infections. And I just want you to think about it as to why they're not useful, and write your answer. Again, you want to write all of the answers for all the 5 questions on a same homework sheet, and then upload it to the Canvas. So with that, this lecture is complete. Let me know if you have any questions. You can email me, and I will see you guys on the 4th in class. So the lecture on 4th will be in person in class, as we meet regularly at Equity Hall, room 132. I'll see you all there. Bye.